The overall goal of this procedure is to observe the effect of specific treatments of interest on cell migration in a three-dimensional wound-associated fibrin scaffold.
This method can help answer key questions in the wound healing field about how alterations in the fibrin network structure influence cell migration into fibrin clots at wound sites. The main advantage of this technique is that it provides a simple and reproducible method for analyzing cell migration within fibrin clots in 3D.
Generally, individuals new to this method will struggle because transferring the spheroids can be difficult.
Begin by warming a vial of frozen neonatal human dermal fibroblasts in a 37 degrees Celsius water bath. When the cells are just thawed, collect them by centrifugation and resuspend the pellet in fresh neonatal human dermal fibroblast medium at a 1 × 10⁶ cells per milliliter concentration. Then seed the cells in a T75 culture flask for a 72-hour incubation at 37 degrees Celsius and 5% carbon dioxide. When the culture reaches 80% confluency, detach the cells with 5 milliliters of trypsin EDTA. After 5 minutes at 37 degrees Celsius, neutralize the trypsin with 5 milliliters of warmed medium and mix 40 microliters of cells with trypan blue at a 1:1 ratio. Reserve a 10 microliter aliquot of cells for counting. Collect the cells by centrifugation and resuspend the pellet at a 1.25 × 10⁵ cells per milliliter concentration. Next, plate the bottom of a 10 cm Petri dish with 5 milliliters of sterile PBS. Then invert the Petri dish lid and add multiple 20 microliter drops of the cell suspension.